Hi guys, this is our project, CIALLO-agent, LLM-driven synthetic circuit compiler. And here is our group members and the contact email. First, let me introduce our project. Our project is an LLM-assisted design pipeline for synthetic biology that bridges high-level user intent and low-level biological implementation using Cello v2. Users can describe a desired biosensor circuit in natural language, and the system automatically generates Verilog input and output JSON files and the compatible UCF configurations required for Cello execution. By combining official biological libraries with automated validation and execution, our platform improves design reliability and makes synthetic circuit problem typing more accessible and efficient. This slide shows our clearest technical example, how we bridge natural language requests with Cello-compatible biological design. First, the user provides a simple prompt. Next, our system translates this request into Verilog logic, where the biological behavior is represented as a logical function. For example, output equals input 1 and input 2. Then, we perform biological mapping, where abstract logic nodes are matched to real biological components. After that, the pipeline automatically generates all required files, including Verilog, input JSON, output JSON, UCF files, and summary files. These files are then passed to Cello for validation and execution. In our experiment, Cello successfully reached the logic synthesis stage and executed the algorithm, which confirms that the generated design is valid and executable. The key takeaway is that our system does not just generate text or code. It produces real biological design artifacts that can work with an actual synthetic biology compiler. This is our web page. On the left side, users can choose different working modes depending on their design needs. For example, freeform request, here it is, allows users to directly describe a biological circuit in natural language, while paper-assisted draft uses a research paper as a supporting input, and custom components is used when users want to define specific biological parts manually. In our project, we mainly focus on the freeform request mode, which is the most directly LLM interface workflow. In the central panel, the user selects the official biological library, such here, like this, and I selected Run Cello and then Generate Design button. Yeah, here we see the results. The pipeline automatically generates structured design files, including Verilog. Here it is, Verilog, and then input output JSON files, UCF draft, and here is the output files, and also we have the summary here. Here is a demo shows the full workflow of CIALLO-agent, from a design prompt to the final dynamic simulation result of a biological circuit. First, the user describes the desired circuit in natural language. This description is used as a prompt input to the agent. Based on the logical description and the input and output device descriptions in the prompt, the agent generates a Verilog file, an input device file, and an output device file. These generated files, together with selected UCF library, are submitted to the Cello. Cello then automatically performs circuit synthesis and simulation. After Cello finishes, it generates many intermediate files. Among them, the netlist file contains the structure information of the biological circuit. For dynamic simulation, Cello first uses the netlist extraction module to extract the biological gates used in the circuit, the connections between the gates. And next is extract biological gate parameters. Based on the cleaned netlist information, Cello extracts the corresponding Hill function parameters from the UCF library, the input device library, and the output device library. Then build and run the dynamic simulation. The Cello simulation module uses the extracted parameters and circuit structure to generate an antimony script. Then it runs the dynamic simulation using Telerism. Before running the simulation, the user can modify the simulation settings in the build file, including simulation time, biological gate time constants, input signal shapes, and input transition times. After the simulation is complete, the simulation module outputs both CSV and PNG files. In this picture, we can view the simulation curves and check the dynamic behavior of the circuit. This simulation shows the dynamic response of the system of two input signals, X1 and X2, and how the final output Y changes over time. The smooth S-shaped increase instead of an immediate jump reflects real biological dynamics, because gene expression, protein production, and reporter accumulation all require time. This shows that our design not only satisfies the logical function, but also demonstrates the realistic biological behavior.